Thank you very much, Thomas. I will share my screen. Let's try and do that first. Yes, here we are. Okay, thank you for the very kind introduction, Thomas, and welcome everyone. Today I will speak about the importance of collecting data on marine megafauna within the EEZ waters. We take a look at both French Guiana and Suriname. So my presentation is going to be first focusing very briefly about what are marine megafauna and why is it important to collect data. Then a little bit about the complex situation of ocean currents and maritime zones. And then we look at what we have on current knowledge regarding marine megafauna. But then we come to the fun part, where I will introduce you some marine mammal species. I will tell you a little bit about their facts, and I will play you some sounds that they produce underwater. And then we'll look at some maps based on the latest information on marine megafauna in Suriname and French Guiana. I will do the same for sperm whales and baleen whales, but also spinner dolphins. We have a few slides on Avi Fauna. And then I conclude with whale sharks and giant manta rays. I will then explain to you the importance of species richness maps to point out places of diversity. And finally, I conclude on the importance of databases and especially international databases. So what are marine megafauna? Basically, to put it simple, anything you can see with the naked eye at sea. So for example, seabirds, because although they fly in the air, they do take their food from the sea. Large predatory fish, think about large tuna, for example, or sailfish. Marine turtles, whales and dolphins, but also squid or jellyfish, and sharks and rays. And for my lecture, I'm going to be focusing on seabirds, turtles, cetaceans, that are whales and dolphins, and sharks and rays. So why collect data on marine megafauna? Well, it's important to understand the impacts of increased anthropogenic pressure in the ocean, as this is a global issue, we all are aware of this, but also these pressures are increasing in the Guianas. I think about oil exploration, fisheries pollution, and and pollution. So we need to successfully manage our seas, and for this we require an adequate knowledge of the species that thrive in our waters. We need to assess the potential interactions that marine megafauna have with anthropogenic activities. So there is an urgent need globally for ecosystem-based management. So rather looking on one species, we are now looking at the whole ecosystem. And we can do this through integrated marine spatial planning across international borders. And I will talk a little bit more about marine spatial planning in a minute. So marine megafauna are iconic members. We all know them from when we were small. Turtles, sharks. Yet we know very little about their distribution and abundance, and also very little about the functional influence they have on the ecosystem. And we do know that their populations have declined drastically due to direct exploitation, think about whaling in the past, but also the ongoing direct take on sharks for their shark fins. But also we have a lot of bycatch in fishing gear, so-called incidental mortality. So the present work explores how we can combine existing data sets into one big database and how we can then best analyze this as well. So starting with maritime zones, we can start on the left. You can see the coast. Then up to 12 nautical miles from the coast, we talk about the territorial seas. Moving on up to 200 nautical miles, this is the EEZ, the exclusive economic zone. Now for Suriname, this is a little bit different because it has been extended. That is because the Sea of Suriname is relatively shallow because of the presence of a plateau. And therefore the EEZ has recently been extended due to an extended continental shelf up to 350 nautical miles from the coast. And beyond that, you have the international waters or the high seas. So here we have a chart showing you Guyana, Suriname, and French Guiana. And together, these are the Guianas. And it's not just on land, but also at sea. And you can see what is quite evident, that starting towards the west of French Guiana and then well into Suriname, you see this lighter blue plateau extending very far offshore. And that is the Demerara Plateau. In yellow, you can see the EEZ lines. And in red is the extended bit up to 350 nautical miles offshore for Suriname alone. So, sorry, I go back one. You see all these little blue wiggly lines, those are the depth contours. And if I take all those depth data, for example, into a software program called GIS, which is a mapping program, then I can also calculate how steep the slopes are underwater. Because underwater, you have hills and valleys, just like we have on land. And here you can see the steeper the slope, the red the color is. And you can see the northern edge of the plateau clearly defined as having very steep slopes where it suddenly plunges into the depth of the high seas. And also of French Guiana, you can see the continental slope there has some red in it. If you compare that to Guyana, you can see that there is a gentler slope there. So regarding the currents, this is a complex system actually. We have the North Brazil current, mainly going from east to west, and we have the North Equatorial counter current going from the other direction. And then these two mass currents, they bump into each other. And that is actually happening right here off the Guianas. And this interaction causes the creations of huge gyres and also smaller eddies. So large circular currents. And these are formed and shed in the Guianas, and they travel then up in the direction of the Caribbean and also into the Gulf of Mexico. So here you can see some gliders. These are pieces of equipment that they throw overboard and they can glide on the surface, or in case of the buoy on the left, they actually sink and they come up to the surface at regular intervals. And they have a satellite transmitter, so we can actually track them. And here you can see on the right the track of all these gliders, and you can see those huge loops where the gliders have been following the gyres. And this then disappears slowly, like I said, in the direction of Trinidad and then up north. Well, how does that look like at sea? Well, this is a front, basically where two bodies of water meet. And in calmer weather, like here on this slide, you will see a bit of a silvery line in the water. If it is rougher, then actually usually this front is rougher. So you have a lot of white caps of water along the front. And these two bodies of water that are meeting are reluctant really to mix. So you can get all kinds of concentrations along the line. And that's where nutrients are building up and where little fish come to take advantage of the local productivity, and they attract bigger fish and predatory fish, and in turn whales and sharks and seabirds. So in 2017, we created a marine spatial planning project. This was launched, like I said, in 2017 by the EU for both Guyana and Suriname. And if I Google what is marine spatial planning, then this is a process that brings together multiple users of the ocean, including energy, industry, government, conservation, and recreation, to make informed and coordinated decisions about how to use marine resources sustainably. So MSP generally uses a series of maps to create a better picture of the marine area, so we can identify where and how an ocean area is being used and what natural resources and habitats exist. So my role within this marine spatial planning project was to deal with marine megafauna data. And this was going to be a little complex because really we didn't have many sightings. We knew that the ocean currents had a rather complex and dynamic character. So we're going to expect spatial and temporal variations. And typically an equatorial tropical sea will have temporary hotspots. And a temporary hotspot is basically a place where suddenly all conditions are right. So we have this buildup of nutrients that I was mentioning before, which is attracting fish, bigger fish. So a lot of marine megafauna make use of these hotspots for feeding, but usually they disappear, these hotspots. And the next day, the marine megafauna will have to go and find the next hotspot. So what is the situation in French Guiana? So we have an advanced data collection already with systematic surveys carrying out on a regular basis, both aerial surveys from airplanes and also boat-based surveys. They have also put together the red list for the IUCN of the species that are most threatened in French territorial waters. And for this process, they also requested data from Suriname and Guiana, because they can then better interpret what is happening across the border and interpret their own data better. Because marine megafauna, they are not aware about the place of the EEZ and all the borders, they just move across borders. So here you see a sample of a boat-based track carried out in French Guiana. And so you can see the lines are the track that the vessel was making. And then you can see in pink those areas which were carried out, the observations. So basically you have two observers on watch and they will look out for marine megafauna. They write down when they start, they write down when they stop their observations, and they write down what they have seen. When conditions are too rough, you can see in black those periods that they were not on effort. And in blue, this is also interesting, this is where they were towing a hydrophone. So that is like an underwater microphone recording the sounds that marine mammals produce. And we will hear some sounds later in this presentation. So what did they see during these surveys at sea? I've plotted here their sightings. Their names are in French. I don't speak French, but I can more or less make up what species they were seeing. But just looking at all the colors and the shapes of the symbols, the diversity is quite high for this region. And what do we then know about Guiana? So Guiana, we're only just started. I mentioned this already. We've done a literature study. We've looked at strandings. We are now searching for data sources, and the best data sources are those collected by marine mammal observers, MMOs, literature, and also online databases. Then we will have to compile the sightings and start building our own database. In Suriname, we're a few years ahead. You can see we started building up the database in 2017. And every year you can see there are more and more data imported to the database. There was a huge peak in November 2019 with seabirds. That is because we were getting permission to use a database that already existed where all the bird sightings were stored for Suriname. So this was a huge improvement on our knowledge of seabirds along the coast, but also at sea. And at the moment, we're also busy to introduce all turtle nesting records that we have of Suriname. So in total, we have more than 10,000 records for Suriname. But it is important that we realize what we are looking at. Like I said, the data collected often by marine mammal observers on board seismic research vessels. And this is a little drawing showing you a seismic vessel, and it's towing 3 streamer cables behind it. And what they produce a very loud underwater noise, which is directed directly downward to the seabed. And these loud noises reflect from the seabed up to the surface and are then recorded through acoustic receivers in the streamers. And the characteristic of the echo of these sound waves will give information whether there is oil presence in the seabed. But you can imagine that for a marine mammal, it is going to be potentially harmful to be very near such a loud underwater noise, because marine mammals are very much reliant on communication skills. They also use sound to find their food. So they've got very sensitive hearing. So therefore they place marine mammal observers on board these vessels. And I have worked myself as a marine mammal observer as well, in order to keep a lookout. And when marine mammals are close to the vessel, we will delay any operations. So typically for about 50% of the time that a seismic survey vessel is at sea, it is going to be in operation. The other thing about the data that we're looking is it's very much dependent on observer experience. You can imagine that a marine mammal observer for the first time going at sea does not have a very well trained eye yet to detect all these animals. So it is important to take that into account as well, because a person who's been at sea a lot, like myself, will automatically make detections much quicker. We have also used data from citizen science, both in French Guiana and in Suriname. Here you can see a whale shark. I have another picture slightly better. But these were seen from a drilling rig back in 2015, and we were able to add those observations to our database. There's also working together with fishermen. So, for example, back in 2013, a trawler at sea recorded a mother and calf humpback whale. And this was an important record. It shows that the Guiana waters are perhaps a corridor for humpback whales, females with calves, to move between their breeding ground, which is the Caribbean, and their high Arctic feeding grounds. So these are whales that move around quite a bit. So when you're collecting all this data, you also need to really do a thorough quality control. And we rated the data that were submitted to the database in the following way. So we have definite records. Those were sightings that had a photograph, so we could actually prove the identification of species. We have moderate rating for records that have a good description but no photo, but they came from a trusted source, so an experienced observer. And poor and discarded data were data without a description or a photograph, or even sometimes looking out of the ordinary. And you can see here a photo of pantropical spotted dolphins in the distance, and you can then see how difficult it actually can be. The dolphins are more or less hiding behind a large swell, and the lighting here with the sunshine makes these dolphins look almost completely black. So I can really only tell because of the very narrow dorsal fin and the beak, and by tuning into the photograph, that these are pantropical spotted dolphins. But for an untrained eye, you can see how difficult this can be. So when we then only take our definite and probable records, we can plot them. And here you can see basically dots on a map. So the position of where these data were recorded. And you can see the case for French Guiana is very much advanced. In Suriname, there are still quite many holes, especially also in the bit of the EEZ that has been extended, because we haven't really been doing much survey work in those areas. These are all whales and dolphins, and this one is all the birds, or Avi Fauna. And you can see again of French Guiana, a lot more research has been carried out, basically mainly through these aerial surveys. You can see a lot of birds along the coast, also in Suriname, but also quite a lot of birds at sea, foraging. And here are the turtles, and it's a bit of a different situation in French Guiana because they have also islands very close to the coast and the water is more clear, while in Suriname and Guyana the waters are more muddy for a longer period and start to become green about 50 kilometers from the coast. So these were all turtles. Mapping marine megafauna. So we just looked at a distribution map. Basically, it shows where a species has been seen in a certain area. But like I said, there are blank areas on the map. Does a species occur there, or do we not know this because we haven't been looking there? So it is unclear how much research has been carried out in a certain area. Also, the data are systematic or opportunistic. And systematic data are collected by observers that make note of when they start their watch, when they stop, and how many animals they saw during their watch. So they can then later compute, for example, how many whales were seen during an hour of effort, or how many whales during 100 kilometers of trek at sea. And an opportunistic record are, for example, those records made by those oil rig workers. They see a whale, or a whale shark in this case, they make a photo and they submit the record. But for the rest, there is no more data affiliated with such an opportunistic sighting. Here you can see systematic survey tracks. These were surveys I did myself of Suriname. So we were leaving in Trinidad and then traveled through Guyana waters and started research in 3 different places. So when you want to study the distribution and density of marine megafauna, you do a series of steps for the Marine Spatial Planning project. So we've looked at the distribution map. And the next analysis would be making a presence and absence grid. We would like to map where we've done systematic surveys and what animals were seen. Interesting, especially also, is a relative abundance map. Like I explained before, you can then calculate an index of how many whales per systematic survey were seen in certain areas. Then you can compute a density or heat map where we plot observations and weigh them on the distance between records. So that is showing you a hotspot, really. Then the species richness, where you plot for an area how many different species occur there. So it gives you a measure for diversity. Then also important is finally using all these maps is making like what I call a sensitivity map per species. Basically showing you the area which we think, based on all the information, is important for a certain species. And the final analysis is what we're doing now in Suriname, and that is defining areas of biological importance. Because when you want to start your project offshore, whether you want to go start fishing in a certain area or where you want to go look for all the oil or drill for oil, it helps to know which species one should take into account when planning a new project and when you need to carry out an environmental impact assessment report. So now we come to the fun bit. I will talk to you about sperm whales, baleen whales, and spinner dolphins. So let's start with sperm whales. Here you can see a female with a very small calf swimming alongside her. This one's really quite small yet. And this is really what we see in Suriname waters, larger groups of so-called nursing pods of sperm whale females with their young. Now, a sperm whale is the largest toothed whale. The males can get up to 18 meters. The females are a lot smaller, up to 12.5 meters. And they can stay underwater for more than 60 minutes at a time. So they can really hold their breath. And when you see them at the surface, like you see in this little diagram, they're really resting. They're really actually taking a deep breath. and multiple breaths. So finally, after 10 or 20 minutes of resting at the surface, it will arch the back and it will fluke up and it will go straight down. It's a bit built like a submarine, really. Here you can see one underwater together with a diver showing you the size of the animal. And this is when they sometimes are resting just underneath the surface. So sperm whales produced a clicking sound, which we call echolocation. And on the left, you can see a sperm whale producing clicks, like sound waves. And these sounds, they bounce back off a potential prey item. And the returning sound, so the echo, is actually what reaches the sperm whale and will give it information. How far is this object? Is it potential prey? Can I eat it? And then it will need to fine-tune where this prey is. So it will actually increase its click train. And that is really to fine-tune and find the potential prey. And in this case, we look at the spectrogram. So you have the time on the x-axis, and it's plotted against the intensity of sound or decibels. So we're going to listen, and we'll first hear the clicks, and then we'll hear this fine-tuning. But in the end, it decides not to eat it because it will start producing normal clicks again. Let's have a listen. So those were the sounds of a sperm whale, and you can immediately understand that it helps when it's not too noisy in the sea for a sperm whale to find its food. So what do we know about sperm whales in the wider Caribbean? Well, there is a website called Aqua Maps where they have already plotted all the information available for the Caribbean Sea, and they have exploited this or expanded this to the wider area using the habitats suitability index for sperm whales. So showing you on this map now where they think the sperm whale may also occur. So you can see the Guiana basin is important for sperm whales. And what do we have now? Here you can see the sperm whales plotted on the map in the Guiana region. And in yellow, the situation for Suriname. And the larger the yellow circle, the larger the pod size of the sperm whales. So you can see up to 20 animals in one group. And also the next map is a presence-absence map. So in gray, you actually see where systematic surveys took place in the Guiana region, in Suriname in this case, and in black where those areas where sperm whales were recorded. So it gives you a better information on presence and absence, basically. And this is a density map. So this shows you more the hotspot areas. So the darker blue, the more records of sperm whales were made. And probably these two hotspots that you see are connected. Like I said before, there is still a lot of holes in our data. And this is a relative sightings map. So here I've plotted how many whales per systematic survey per grid cell. So the higher the rate, the red the cell is. So it's, of course, resembling the two hotspots that I showed you before. And then we can draw up a consensus map of known and probable occurrence. We know, for example, that sperm whales, they like steep slopes underwater. That's where they can find their food. So we can assume that the Demerara Plateau as a whole is going to be important, but we see a concentration of records on the west side of the plateau. There's still many question marks, so there is still need for further research. If we move to baleen whales, let's start with humpback whale. Humpback whale can be also up to 18 meters in length, so a good-sized whale. And unlike the sperm whale, it doesn't have teeth. So it has baleen plates, and these hang from the upper jaw. and it uses these baleen plates to filter out food particles. And these food particles are, for example, krill or small fish. So they can be very acrobatic, humpback whales. They have huge long flippers. And here I have a collation of photographs. So they frequently jump out of the water and they really expose their flukes as well. And in this case, you can see a completely white fluke. They can also be completely black or any type of variation to the degree that we can actually recognize individuals too. Now it's the males that make the underwater sounds. They sing in order to attract the females. So let's have a listen. So pretty impressive underwater songs there. So what do we know about humpback whales in the wider Caribbean region? Well, here you can see the breeding ground plotted. Again, this was Aqua Maps website. But of course, it doesn't show the migration routes because these whales, like I mentioned, they are going all the way up to the high Arctic for feeding and come back in 6 months' time for their breeding process in the Caribbean, to the breeding grounds. This is a map for the Bride's whale or Broodies whale, which is a tropical baleen whale and is likely to be present year-round. And it's quite interesting. They have plotted suitable habitat here. And you can see the outline of the Demerara Plateau pretty well there. So I have to pull all the baleen whales seen in Suriname because our sample size is not quite high yet. But in red, you can see the sightings of baleen whales in French Guiana, and you can see they're concentrated on the slope, the continental slope. And in Suriname, you can see it's more confined to the Demerara Plateau. So if I make a kernel density, then you can see indeed there is a larger hotspot on the Demerara Plateau, but also I have the feeling that the continental slope, especially on the half near Guiana, that bit of the Demerara Plateau is important for the baleen whales. So if I make a consensus map, it becomes a little bit more complex because in green you can see the potential migration route through Suriname waters. But in orange, you can see this corridor that I was talking about for female with young. And in pink, you can see the area where we've had most records and where we have seen them feeding. And in blue is what I meant with the continental slope area, specifically on the Guiana side. I think that's where more research should be carried out. Moving on to spinner dolphins. A very acrobatic small dolphin with a very long beak and a little black tip right at the end. They are highly acrobatic and they really show spinning behavior, and it's the only dolphin that can spin up to 7 times. So I have a few photographs here of spinner dolphins. You can see that long beak. They frequently approach the vessels, like in this case, to bow ride and leap out from the water. Here you can see a photograph showing the spinning movement. You can see the water is flicking off the body. And spinner dolphins, they make very high-pitched whistle sounds and echolocation. So the whistling might not be audible for everyone, but I'm sure you can detect the echolocating bursts. So I hope everyone could hear that. I made also a consensus map for known and probable occurrence of spinners, and you can see especially the western side of the Demerara Plateau. Still many question marks though, and again there is much need for further research. If we move on to Avi Fauna, here you can see a Cory's shearwater, which is a pelagic seabird. It doesn't come close to the coast. It actually breeds on the other side of the Atlantic and also in the Mediterranean. But here in pink, you see an area where we've seen Cory's foraging, and mainly in areas where we have sargassum. So that is a floating seaweed that can build up to certain times of the year. In blue, though, probably they will use the whole area and also Guyana and French Guiana and also the international waters. If we look, for example, at magnificent frigate birds, which you probably know better, then these are more confined to the coast, but they will also go offshore for feeding purposes. The brown pelican, however, is really confined to the coast. And here I have plotted the data, made density maps as well, and I'm able to show those areas where we have the most records. And moving on to whale sharks now, we documented the first whale shark in Suriname in 2013, and it's the largest fish and the largest shark in the world. Up to 10 meters in length, you can see the diver is being dwarfed by this huge shark. Luckily for the diver, whale sharks are vegetarians. So they filter the water. They don't have baleen like baleen whales, but they have gills, which they use to filter the water. And then they feed on small particles, fish eggs and fish larvae. Now here I have a small video clip that was taken by the Suriname Coast Guard. And you can see parts of the whale shark. So the whale shark is blue with white dots. Let's have a look. So at no time you can see the front and the back of the animal, but it is definitely attracted to the boat and swimming alongside it. So a stunning record from the Coast Guard in Suriname. And when we plot all these records, we can see there's a good concentration of records in French Guiana. We're slowly building up the database in Suriname as well. We've got also hammerhead sharks, for example, in green, and the whale sharks are in blue. Making a interpretation map is difficult. It's very hard to detect sharks and rays when you're standing on a boat. In French Guiana, it is easier with the aerial surveys to plot those animals when they are swimming just below the surface. But it seems that the Maritime zone of Suriname is more important for sharks than further offshore. The giant manta ray, I'm sure you are all familiar with this giant as well. It is the largest ray on the planet. It measures up to 7 meters from wingtip to wingtip, and they often also raise the wingtips from the water, as you can see in the photograph on the right. They are also vegetarians, so they use their gills to filter out small particles, fish eggs and fish larvae. Quite a few records in French Guiana, the little red triangles, and in Suriname, I've plotted them to species level, but the giant manta ray is really a species that we see in the shallow region, and that's probably the same also for French Guiana. Further offshore, we have the Chilean devil ray as well. So if I then make an interpretation plot, then we see that the concentration is really in the Maritime zone, but we also have an offshore foraging ground for rays. So moving on to marine mammal species richness maps. So again, on Aqua Maps, they have made one for the wider Caribbean region. And per grid cell, they have predicted how many species would occur in these areas. And we have up to 22 species even on these maps. It's pretty impressive. And if I do that for the Suriname data, here I've plotted cetaceans, turtles, sharks, and rays. We have up to 11 species on the Demerara flank, on the west flank. So the darker the cell, the higher the number of species. If I do this for pelagic seabirds only, in this case, the bluer the cell and the darker blue, the more species we see, especially the coast is important as we could predict, but also the Demerara Plateau shows good numbers of species. If I pool everything, so cetaceans, turtles, shark rays, and also pelagic seabirds, then we can already reaching up to 21 species per grid cell. And this is a brilliant tool to show you those areas where the highest diversity is at sea. So now coming to the end of my presentation, I have a few slides about the importance of databases. This is a quick slide showing all the seabirds plotted for the wider Caribbean region. I'm currently analyzing these, and in particularly also the yellow plots on the east side of the Caribbean Sea. But it shows you how much more you can do when you have more data. Seabirds, they also migrate all over the world. They come into the area. You can see where are the foraging grounds, which are breeding grounds. How do they move between certain areas of the sea? Marine fauna are highly migratory. And when you start off as a marine biologist and you want to collect data, then you will develop this often for a single region and also for a short period of time. And your data are just not building up high enough to do proper analysis. So these data must be brought together on a global scale, preferably, and these databases should be openly accessible. And one such database is OBIS CMAP. Now, OBIS CMAP stands for Ocean Biogeographic Information System, Spatial Ecological Analysis of Megavertebrate Populations. So you can see the map. They already have a lot of data collected worldwide. There is a little green cloud there in the place of French Guiana, and they have already put their data on OBIS CMAP. We have, for Suriname, also submitted the data, but they are currently being reviewed before going public. So any minute now, hopefully, we will see the records for Suriname too. As a data user, you can go to OBIS CMAP, the website, and you can look up locations and dates of species. Data providers still hold the ownership. The user needs to agree to terms of use, and basically you need to get permission when you want to publish the sightings. So it's a very easy system, and this international database holds data collected during ship and aerial surveys, but also satellite tracking, for example, turtles at sea, acoustic data, those underwater vocalizations that I was playing, and data also on sea turtle nesting and photo ID and model data. So I show you a little map of Guyana and the data that is currently in OBIS CMAP. So it's mainly sea turtles, but also a pod of sperm whales. And this would be a start for building up a database. There are more similar databases where we can look into. Here you can see the situation for French Guiana and a little bit of overspill into Suriname, but hopefully the data for Suriname is going to be public very soon as well. So I found it was very easy to import data into OBIS CMAP. There are also options for restrictions. You can also hide your data, but it is also a brilliant tool because you can put your data somewhere safe without the risk of losing it. You can collaborate with fellow scientists, and there is a communication channel between the person who wants to analyze the data and the person who has collected it. You can associate your data with oceanographic variables. Think about sea surface temperature, chlorophyll levels. You can make time series, seasonal graphs, and this all is helping towards marine spatial planning. So where are we next now? We will continue for the Guyana region to access old data, but we also need to make new data. We will build up awareness and hopefully get more citizen science records where we involve the public. And we can have to start carrying out dedicated marine megafauna surveys, and university studies can be a big part of this. We can build up our online databases and also submit data in the international databases. So, only an integrated cross-border database is going to improve our knowledge and will aid environmental impact assessments that are so much needed for offshore activities. We can then identify our areas of biological importance, that's what we're doing now, evaluate potential threats that marine megafauna face, and describe different marine zones and habitats. And then we can use all the different information for a rapid assessment approach to identify and prioritize those areas that are in need of conservation. And thank you very much for attending my presentation. I hope you have learned a little bit more about marine megafauna, and I will hear from you if you have any questions. I'm going to stop share my screen now. Yeah, I think I...